to this muscle contraction class, that is contraction of skeletal muscle. Here you need to know a few basics, the microanatomy of the muscle, how it contracts, and a few of the changes that are happening in the skeletal muscle while it's contracting. Okay, so as we can see here, this dark band is A band, these two bands, they are A bands, and in between them sandwiched is the I band. Okay, and easy to remember how is A band is anisotropic, it will not allow light to pass, that is that means it will appear dark. So DARK, highlight this A, dark is A band, and isotropic will allow the light to pass, so light, isotropic, highlight this light zone, dark and light. Light zone is I band. Okay, and in the middle of dark area is this M line, and middle of light area is this Z line. Okay, next let us go into the details. So here when you see this, this is the I, right, I band, these two are the I bands. And this is the A band, right, dark A band, anisotropic. Good. Then what is this? This is the M line, as you have seen. These are the myosin filaments, and this is myosin molecule, and this is the actin filament. We will deal about the microanatomy of the actin in a while. And what happens is these molecules will bind to the these myosin molecules, they'll bind to the actin, they move inward, see here, they move inward, and the actin, this actin moves inside like this, and the muscle contracts. And within this dark zone there are few light areas, right? So what is it? The center is the M line, to which the myosin filaments bind. Adjacent to M line is the pseudo H zone, and by the sides of pseudo H zone is the H zone. So what is this H and pseudo H? This dark area is due to presence of myosin, right? And here there is no myosin molecule, there are myosin molecules but the interlink is not formed, and here actin myosin crossovers are never formed in this pseudo H zone. This light area is due to absence of myosin molecules, and this total light area is due to, that is white, it is due to absence of actin myosin cross bridges. Okay, remember that this pseudo H zone is due to absence of actin myosin cross bridges. Okay, fine. We have seen this. This is the Z line, right? It is present in middle of the I zone, and this is the M line in present in middle of the myosin, that is the dark zone. Very good. It is easy way to remember, this M line is in middle of myosin, and remember this as a small city, and away from city is the zoo. So Z line is outside the dark band, M line is middle of the myosin. Okay, this actin, let's go to the important structure of actin. In this actin, consider this is the Z line and this is the actin. Okay, there are two chains of actin and there are two chains of tropomyosin, okay, covering the actin, around the actin. Now you'll ask me, why to cover, right? Simple tendency to ask anyone who says that the actin is covered. Let us see that. On the actin, on both the chains, there are few active sites. Okay, these are the active, for suppose these are the active sites to which the myosin molecules have to bind to initiate the stroke and contract, isn't it? They bind, move inside, and the muscle contracts. Okay, in order to prevent this, these tropomyosin chains are covering the active sites on actin. And so this is the actin, right? Let us write. We have seen here in the previous slide, I have written that there are this one ACTI. I highlighted this. Why? Because there are three proteins. Okay, for say these are the three proteins. These are the proteins of troponin. Okay, they are CTI. They are present on actin. So ACTI. This is a simple, very simple to remember. Actin, within actin there are troponin proteins. They are CTI. So what is the importance of these proteins? So you already know that calcium is required for muscle contraction. So how is this calcium making the muscle to contract? So as soon as the calcium is released, it binds the C protein of troponin, which causes the contraction of tropomyosin, exposes active site which is bound by myosin molecules. Okay, it's good. Until now we are good. Okay, this is the sequence. What happens? See how it contracts. So as this is the actin chains, this, when calcium is bound to this protein, this troponin proteins are present intermittently over the tropomyosin and actin chains, actin filament. So when the calcium binds this C on troponin, this will cause the chain to contract. That means as of now the tropomyosin chains will become like this and expose the active sites. Okay, to which the myosin molecules will bind and they have to move inside. How they are moving? So they need some energy, right? So how the energetics work here? Let us see into that. So when the myosin filament, myosin molecule binds to the active site, right? This binds to the active site and moves inside. Till now this myosin molecule do not need energy because this already has an affinity to bind to the active site, so it does not need energy. But after binding and moving the actin filament inside, it has to release, right? So binding, stroke, release, again bind, stroke, release. So for the stroke, till the stroke you need energy for stroke and to release also. So what happens here? At the while it has to release, it has to be released from the active site. Here this myosin will cleave the ATP, myosin head will cleave the ATP to ADP plus PI. So this yields energy for release of myosin molecule from the active site, right? And again this binds to the active site, to the next active site. So after binding, the stroke is happening. So you have to remember what? You have to remember this one very good thing. The energy required for the subsequent stroke is already stored in the head of myosin, so it doesn't need to cleave anymore. Okay, next, coming to a simple thing. So this is M line, this is Z line, this is myosin, this is actin, this is myosin, right? So let us see. So this is the I zone, this is the M zone, sorry, A zone, A band, this is the I band. So what happens when the muscle contracts? This actin will move inside, right? Because myosin is what that is making the actin move. So this will move. When the muscle contracts, what happens to the I band? I band decreases in size. A band remains constant. And what happens to the pseudo H zone and pseudo H zone? What happens? PH zone remains constant because there is no actin myosin cross bridge, and H zone might decrease. Okay, H zone may or may not decrease depending upon the strength of contraction. Full contraction, H zone will diminish or disappear. And this Z line may touch myosin. Okay, but Z line never touches M line. Okay, never touches M line. These are the few important points you have to remember. So that is how the muscle contraction and coupling takes place, and how there is a release of head, and what are the zones that are diminished in size and constant in size, various molecules and molecular level physiology that you have to know. That's all. Thank you very much.